Joanna said, should she give me an introduction? I said, I can introduce myself, I suppose. But that's, excuse me, that's not the first point to make. The first point to make is to introduce Sorenza. So Sorenza Bastiaens is a PhD student in my group. She is, without exaggeration, one of the most talented scientists, young scientists I've had the pleasure of working with. And she really understands neural mass modeling, which is why she's in the schedule. Unfortunately, her flight was delayed, and she had a connecting flight, which meant that she couldn't get here in time for the talk. So she will be joining us in the afternoon, and please everyone kind of say hi to Sorenza and have a chat with her when she gets here. So I'm kind of stepping in, and I'll do my best to allay the disappointment of me not being Sorenza, but I really want to defer all of the credit for the presentation and so on to her, because she's done all the slides, so it's kind of minimal job for me. Great. So the aim for this part of this final session, I'm going to actually just pop to here, of the morning. Actually, just quick recap on the plan here. So we've had our first two speakers, Victor and Breaky, third speaker on behalf of Sorenza. After this talk at 11, we'll have a 20-minute panel discussion. So Breaky, Vic, you need to be here at 11, FYI, up on the stage. And yeah, we'll take more questions, things that kind of span all of the talks we've had this morning, and whatever you guys are interested in hearing about. And then we lead into the practical session that Davide will lead at 11:20, taking us up until lunch. So that's kind of the plan, just finishing off the morning. And the idea with this talk now is to continue the progression, the educational progression, the introductory progression. That's the idea of the morning session here. It's fundamentals and background, and kind of move it just progressively in the direction of some of the more, say, applied themes and applications that we'll be hearing about in the afternoon. So that's the kind of nominal topic of this session. This talk is to continue with the basic theory, but kind of give you a little bit more physiology. Now, in practice, you've already actually heard a fair bit of physiology in the first two talks, so there's a lot of kind of overlap. But I will be going on that trajectory, and you'll see what I mean specifically on that in a few slides. Yeah, so here's the plan. So I'm gonna do a little summary and just describe a little bit the kind of broader landscape of neural mass modeling and the kind of flavors of modeling and mathematical approaches that are used in the field, many of which you've heard about already, but some of which you haven't. And then take a little bit of time to focus on three specific models just as a kind of demo, effectively. And they're chosen to have some connection with some of the talks that are coming in later in the afternoon. But they're kind of exemplars because it's kind of impossible to cover every model that is used in the field at any level of detail. But I'll talk about some specific models, and then just comment on how these get brought into the kind of whole brain connectome-based neural mass model simulation approach, and give you a little précis of the application talks that we'll be hearing in the afternoon. Okay, so let's jump in. So this is HBM, so you guys have some idea of how neuroscience works. We've got a variety of interesting things at multiple spatial scales, and I mean they're all interesting and vibrant areas of research. We're generally interested in the right-hand side of the slide here. I mean, to be honest, lots of us are interested in everything. It's not necessarily an endearing personality trait. But I would say the right-hand side of the slide and maybe kind of in the middle. What Sorenza quite smartly did here was to not kind of place mesoscopic at any particular level, because Something to bear in mind if you're kind of new to the terminology here is, honestly, so mesoscopic would be kind of somewhere in between macroscopic and microscopic, and exactly where kind of depends who you talk to. And for that matter, where macroscopic lies kind of on spatial scale depends who you talk to. So one person's mesoscopic is another person's macroscopic, and don't be too surprised if you find kind of quite large differences in what the level of spatial scale that these terms are used for. Microscopic, I think, is kind of fairly clear because it kind of pertains to microns, more or less, so that's kind of anchored. So whole brain, we're kind of thinking about these things. And these are the imaging modalities that we're typically interested in, and we've already had a lot of interesting info from Breaky and Victor on EEG and fMRI so far, so I won't dwell on that. Now, this is kind of my key slide for this talk, so you can kind of ignore everything else that I say, but maybe just listen for the next 2 minutes. Because this is kind of trying to kind of pick up with what we've heard some specific examples of, and also some deep analysis of the dynamics in both of the previous talks, and just kind of look at the broader landscape of approaches that are used in this field. And I mean, the field of computational neuroscience, it is. So we have essentially a kind of progression, a continuum, if you like, of more biological to more abstract. Often these are associated with spatial scales, so your most biological neural models are often your smallest neural models, but that's kind of not, they don't map on exactly. But yeah, broadly speaking, kind of following up this progression, so we have, say, multi-compartment detailed morphological models of individual neurons that you would see and kind of use in the neuron simulator software, for example, Henry Markram type stuff, if that rings a bell. And that'd be your kind of most detailed, not necessarily the most detailed, but a relatively more detailed type of neural model. And then kind of, there we go. So we'd have, say, multi-compartment model, then going to, say, the level of an individual neuron. This is quite a common, I mean, it might even be the most widely used representation in computational neuroscience is the cell, and often a cell that spikes. So a spiking neuron is a kind of a simplification in terms of space, because point is a point process, so we don't have, say, a complex dendritic tree, but have some set of equations that describe how a kind of cell behaves. And then we move up to our larger spatial scales, neural populations, as I was saying. So neural population, this is kind of the term that I prefer to use when we're talking about the class of things that includes neural masses, neural fields, mean fields. There's kind of a Venn diagram of overlapping semantics there, but you get stuck a little bit if you kind of commit to any one. So neural population is kind of a general description of these types of models where we're saying this is a patch of tissue, and I want to describe the average activity of this patch of tissue. And that's mostly what we're hearing about here. And then our phenomenological models, again, they're often we use them a lot. They're used a lot for models of neural populations, but they abstract a lot of the physiological detail. And so some of the kind of key examples of these, and this is what I'll be focusing on here. So the left-hand side of the slide, this is just kind of primer on other parts of computational neuroscience that you won't necessarily hear about today. On the right-hand side of the slide, these are things that are kind of relevant to what you're hearing about today. Some of these you've heard already, in fact. So we have things like these neural mass models that abstract a cortical column into a set of neural populations that often has two main players, an inhibitory population and an excitatory population. This is probably the most common general type of neural population model. And there's several that kind of fit that category. There's other kind of variations that I'm going to talk about in the next slide, where we have conductance-based or convolutional-based models, and then we have point processes models, or we're kind of extending into space. These are all in the general area of our neural population models. And then phenomenological models, so the idea here is, I would say about half of what we heard from Victor and Michael in the first two talks was, we're certainly talking about the phenomenology of abstract dynamical systems. In one or two cases, it was then, so like Breaky showed this Breakspear-Terry-Friston conductance-based model. So the things that you can see very clearly in your phenomenological models, often you can see them reasonably clearly in something with a little bit more physiological detail, but the phenomenological models really just focus in on some particular aspect of the dynamics in a way that's kind of suitable for studying theoretically and kind of very clear. So that's our landscape. Okay, you can stop paying attention now if there's other things you want to be thinking about for the rest of the talk. Now let me go into. Oh, I'm having the same problem as Victor now. How did you solve this? You press the button here. The mouse. Okay, good, thanks. Okay, so I want to spend a bit of time talking about neural mass models specifically. As I said, these are one type of the computational neuroscience landscape, but they're an important type because they appear a lot in whole brain modeling and neuroimaging and the work that we're talking about here. So this isn't everything under the sun, but this is it's probably the largest kind of single class of models that you'll encounter if you're like working in this field and reading the literature. And so a neural mass model, essentially we have two general types. We're called convolutional models and conductance-based models. The general difference between these two is that your conductance-based models, which the three-dimensional Breakspear-Terry-Friston one that Breaky was talking about just now would be an example of, those are, a little bit more physiological in the sense of you have terms in there that are often like time constants that represent ion channels. The assumption, kind of theoretical assumption with conductance-based neural mass models is that the behavior of the neural population basically looks like a neuron, right? And this basically what that implies is that the behavior of the neurons in that population are very strongly synchronized and very similar, and that's what motivates the use of a kind of mathematical form that is basically similar to the level of the single neuron, because all the neurons in the population are like very coherent. So that's kind of the idea with conductance-based models. I won't talk any more about that for now. Your second class of neural mass models, or kind of the first or second, the other one, we call convolutional-based models. I'm going to talk about these a little bit more over the next few slides. And these are less physiological, more phenomenological, but also the rationale is quite intuitive. It's quite a kind of intuitive framing for how you might think of the activity of a neural population. So let's kind of go into this. So your general elements of a convolutional neural mass model, there's two. There's what we call a rate to potential operator, I always get this the wrong way around. The pulse to wave operator, which represents the postsynaptic response of the postsynaptic neuron to its inputs. So the way that the convolutional neural mass models are kind of intuitive in the way I was saying is that they basically divide your description of a neural population into these two parts that basically represent the input and the output, like what a population of cells does in response to its inputs, and then how it relays that onto the next step. So that's encoded in our pulse to wave operator, or rate to potential, and our wave to pulse operator, or the potential to rate operator. And what these look like and like how they behave is an impulse response that's in the time domain, basically telling us how fast the postsynaptic membrane potential is rising and then decaying in response to an input. The parameters that are determining the rise and the decay are the amplitude and then the rate constant, the time constant of the decay. So this is the input, this is the response of the postsynaptic cell to its input. And this is typically described in convolutional neural mass models with this kind of convolutional operator that can be described as a second order nonlinear differential equation, which can then be separated out into a pair of coupled first order differential equations. So that's the bit that's giving you the response to the input. And then the second part of the picture for your convolutional neural mass models is this guy here. And this guy is actually kind of everywhere in neuroscience and beyond. What I'm getting at there is that it turns up in neural networks, guys, have you noticed? It's a saturating response, okay? So this is basically saying I've got some input level in terms of a memory potential, and then I've got some firing rate, and then I'm going to have some linear part of this, but then at a certain point it's going to saturate. And this kind of thing, you have an equivalent thing at the level of single neurons. You have the same kind of thing in artificial neural networks. The sigmoid is kind of everywhere in biology and beyond, but it's the second part of our convolutional neural mass model representation. And so what do we do with this? How does this kind of look in practice? Also having the okay. So that was a little deep dive on the structure of a convolutional neural mass model. Now I'm going to go through three specific models, two of which are convolutional neural mass models, and one of which is a phenomenological neural population model, and all of which have some relation to things that you will be hearing or have heard already today. So those three models are the following. A Stuart-Landau model, Jansen-Rit, and Robinson-Rennie-Wright, or just Robinson model. So each of these is, you can see already, we've got two blobs here, but there's basically, I guess it's two-dimensional, so we've got two components there. But these are all relatively simple, right? So we're saying this bit of tissue is going to be described by something that has two variables. In this case, it's 6 variables, but it's kind of 3 two-dimensional variables. Same thing here, it's going to be 4 two-dimensional variables. They're not very complicated models as far as the broad sweep of computational neuroscience goes, but they have a lot of interesting features. So Stuart-Landau model, this is a particular favorite of, well, I assume from what I read, favorite of Gus Deco and colleagues, kind of uses it a lot for modeling fMRI, is somewhat similar, not somewhat, it's like very similar to some other models you'll encounter, such as the Fitsu-Nagumo equations, which for For TVB people, is the generic 2D oscillator. It's kind of similar. And one of the ways in which why it's useful is it's also essentially the normal form of a supercritical Hopf bifurcation. So we can guide the equations for the dynamics of this oscillatory, well, of this state variable through this Hopf bifurcation, where it basically goes from having a fixed point to having a limit cycle, and we can do that as a function of a single parameter. So that's what's going on here. So this is your kind of Stuart-Landau oscillator in the subcritical regime or the fixed point regime, where you can't exactly see, but it's kind of circling eventually back into the middle, and then we move A past the bifurcation point. So this bifurcation parameter crosses the bifurcation point, and then the flows have contracted to a two-dimensional manifold in this space, which is the limit cycle. So it's basically showing a stereotyped rhythmic oscillation at a parameterizable frequency. And then if we add noise into the picture, which we've already had a discussion of from the previous talks, we can generate a kind of equivalent version of this kind of rhythmic activity with some noisy fluctuations, and that actually has a lot of desirable dynamical features. So Stuart-Landau will feature a little bit in talks that we've got coming up from Joanna and from Josephine. I won't comment any more on it right now. Really looking forward to Joanna's talk, by the way. She has some really amazing scientific results over the last few years. Okay, so that was Stuart-Landau. So Jansen-Rit. Now Jansen-Rit, this is one of the real workhorses in the field of whole brain modeling. It's maybe it's like everyone's second favorite neural mass model. I had a philosophy lecturer once who said that about Quine. It's like no one like loves it and it's their favorite, but lots of people think it's kind of a good second best. Anyway, Jansen-Rit model's been with us for a while. I mean, the original kind of essential structure of it has been with us since the 70s, but it was introduced by Jansen and Rit in 93 to 95. So this is a three-dimensional, three-population neural mass model where we've got pyramidal cells, excitatory interneurons, and inhibitory interneurons. Each of these populations has the impulse response for whether it's an excitatory postsynaptic potential or an inhibitory postsynaptic potential. They both have this general form, but we have different parameters for whether so the blue one is excitatory, and the green one is your inhibitory population. And the circuit is kind of like so. So there's basically two loops, an inhibitory loop and an excitatory loop. And these nominally correspond to cell types that kind of live at certain parts of the cortical column. So this general structure, if we lay it out according to, as I was saying before, so we basically have three second-order differential equations that are giving us the dynamics for the convolutional neural mass model here. So we have basically the same thing for each of these. And this has a lot of dynamical structure, like a lot, or might even say too much. A lot of richness, and it's kind of being used a lot. It's a favorite go-to for two main things that you encounter in brain data: oscillations and also evoked responses, kind of EEG evoked responses. We have. Is there another one of these? Like a second one? Or is there a second one of these? Yeah? Is it a bit better? Thank you. Oh, much better. Sorry, Vic. Solved it. Okay. So a lot of people have done some great work on studying the maths of Jansen-Rit model. We as in Sorenza, did some contributions to this recently. So looked at the kind of bifurcation structure of a Jansen-Rit model as a function of its connectivity parameters. And you see the same type of thing that we saw several times, and also what I was showing you essentially with Stuart-Landau. So we have, as a function of one of these connectivity parameters, we can essentially guide the dynamics. So the dynamics being the time series here. So each of these little symbols, these are different points on values of this C3, so this excitatory connection strength. So as we increase this and studying the the eigenvalues of the Jacobian of the linearized form of this differential equation. So we look to see when it moves from being stable to being unstable, so negative to positive eigenvalues, and what the corresponding dynamics look like is the following. So we move into an alpha generating regime where we get this limit cycle oscillation here as a function of our connectivity strength. And what this kind of means, to kind of go the physiology direction here, is if you have some, let's say, intervention that modulates synaptic connectivity or individual differences in patient populations or whatever, where you think there's some kind of difference in the strength of the connections, between these population types in your cortical column, then that would kind of imply that their transition to an oscillatory regime would occur at a different location in terms of the connectivity strength parameter. So all of the, and another way of just summarizing this is a lot of the dynamic stuff that we had already in the first two lectures kind of carries over a lot to Jansen-Rit. But it's more work to do the maths. Certainly is for me. So we have one talk today talking about Jansen-Rit. This is going to be Davide in the afternoon. And as I was saying before, it's been Jansen-Rit's kind of a workhorse for oscillations and for evoked responses. And the work from Davide is actually focusing on the evoked response, specifically the evoked response to a stimulation that is in this recent paper, which is under review, coming from an intracranial stimulation from depth electrodes from epilepsy patients, but also in previous work that's published already, the same thing for TMS with TMS EEG. So we're actually big fans of Jansen-Rit as a kind of good representation of the things we see in TMS EEG evoked responses. Okay, so this is the final final third of the three exemplar physiological models. As I said, I'm calling this Robinson-Rennie-Wright. I got this from, fun fact, I got this from an early version of TVB code that you can find in the commit history, but I think you'll never find it if you don't delve through the commit history. But I dropped the B off, which was stood for Breakspear, but we had reasons for that. So this is a kind of next level, say, addition of complexity in the convolutional neural mass model type. And Breaky was showing some stuff. I think the final things he was showing was with the Fryer work. That was with this model, right? Exactly that model. Yeah. So we've kind of already heard this already. He didn't emphasize so much this bit though. So the kind of construction of the model in terms of the way that it is similar to Jansen-Rit here, right? So we're kind of breaking down the three populations and thinking about them kind of circuit-wise. So the circuit for your Robinson-Rennie-Wright model is this. It's not a cortical column. It's a corticothalamic circuit. So we have some neurons in the cortex, and then basically an excitation inhibitory neural population in the cortex, then a corresponding, well, not corresponding, another excitatory and inhibitory neural population in the thalamus. So 4 neural populations plus a conduction delay. Where's my tau? Somewhere. T minus T0. Plus a conduction delay between the cortex and the thalamus. And the so there's quite a lot to say about this model. I won't go into like much of the details at all, just to say the general structure. So this is another convolutional neural mass model. It has each of the state variables is given by this pair of differential equations that, like, as a pair is a second order differential equation. We kind of break them up into first order ones, and then couple them together between the populations. So we call this the Robinson model, or the Robinson-Rennie-Wright model, by kind of the complete version here. But when you kind of encounter or read about Robinson model in the literature, and when you're talking with people, it actually kind of usually refers to like 2 things. One is this corticothalamic loop. So this particular essentially type of circuit for generating alpha. There's the kind of theory in this model, the kind of big theory, is that this circuit is the thing that generates this oscillation. It's the critical kind of feedback loop that generates other things, but particularly the alpha rhythm. So that's one thing that people will kind of mean when they talk about Robinson model. The other thing that they mean, which we also will hear a lot about, is this thing, which is the wave equation part. So I'll talk about that again to cap off my intro. A little kind of wrap up on the. So final thing about the dynamics here is something that really appeals to me about the Robinson model. Is there's a simplification of it. So the original equations, you know, so I had in the previous slide, some 8-dimensional, 8 differential equations. 8 dimensions is a lot for a lot of things. 3 dimensions, however, is not bad. You can view things. So there's kind of smart kind of simplification of the Robinson model down to a three-dimensional kind of reduced state space where the dimensions of the state space correspond to the essentially the kind of feedback loops within the cortex, the feedback loops within the thalamus, and the feedback loops between the cortex and the thalamus. And most of the dynamics in the three-dimensional space that kind of represented by these 3 reduced state variables, it kind of reproduces pretty much everything that you see in the full 8-dimensional system, which is great, because you can really get a kind of intuitive feel for how this system behaves. So Robinson model, big fan for alpha. And the second part that I was mentioning there, and this is a nod to one of the talks we'll have in the afternoon, but we also had a mention of it from Breaky just now, is the wave propagation part. And this came first. So the kind of Robinson model per se was some new ideas around about 1998 about how to represent wave propagation and kind of macro scale wave propagation in the brain, and then a few years later adding in the thalamocortical loop that I showed you just now to give you these alpha rhythms. But the first part, the wave propagation part, this is what makes it a neural field model. So do I have neural field on the side? I don't think I do. So when I was talking about neural population models so far and neural mass models, didn't really go into neural field models. Neural field model is essentially when you have some spatial continuity in the kind of spatial part, and that exists in this guy here. So the nabla, set of partial derivatives on the spatial manifold. So this guy kind of does a lot of work for how these dynamics kind of behave over space, and especially when you kind of when the system is studied in a regime where it's in a steady state. So in the steady state, you can kind of decompose the nabla operator into these geometric eigenmodes, which are the generalization of the spherical harmonics that are kind of familiar with spheres. So these guys kind of define the basis set that governs the neural dynamics of this system, and in the steady state, they're kind of like standing waves. But they also contribute to the generation of traveling waves in the non-steady state as well. So we'll have a lot more on this from James in the afternoon, and this is a kind of hot topic in the HBM world of late, because there's been a lot of interest in this type of approach to understanding brain activity. So yeah, little kind of précis of things to come, and I'm done with the intro, like I think just bang on time. But just to kind of finish up, so we've got so I gave you a little bit of a breakdown of some other specifics of some neural mass models and neural population models that are kind of workhorses in the field, and gave you a little bit of the landscape of how these kind of fit together. We didn't talk so much about the connectome bit. So there's going to be a lot more of this in the afternoon. And I mentioned this in my initial remarks this morning, but the room was kind of less full. But the general form of our connectome-based neural mass models is these guys we've just been talking about sitting on the connectome, giving us the dynamics that are then going to generate some interesting kind of brain imaging-like neural activity. So that's kind of where it converges to from the point of view of an HBM crowd. And I think that's a good point to end the talk. Well, okay, so final, repeating the précis. So what we've got coming up in the afternoon that are applications of the models I've just been talking about is Davide's work on these TMS-evoked potentials. This is actually the 2023 paper that I was mentioning before. You will talk about this, right? Yeah, so I won't give any more spoilers. I'm biased. And then we have a talk from Josephine Crusat and not Povilas, but Andrea Diaconescu, talking about different clinical applications of these approaches, one in the case of drugs, and the second in the case of psychosis and kind of schizophrenia. So both of these talks and Davide's all will give you describe stuff that is using the models that I've been talking about in the last half an hour. I'll wrap up there. I won't thank myself, but I'll repeat thank you for Davide and Joanna, and especially thanks to Sorenza for preparing this talk, and sorry for my kind of half-assed version of her talk. Also, Sorenza's giving a call out to folks in my lab, so I should definitely repeat that. Some of which, by the way, I didn't mention, are here today as TAs. So can we get a wave? Okay, Parza, Zhang, Minarose, thank you. These guys are going to be helping you out in the practical session. They already helped me out by bringing me a piece of paper and some water from the stage, so they've already kind of earned their keep. But yeah, these guys have all contributed a lot to the work that Sorenza's put together in this presentation. Thank you, and here's some info that I'll just leave on the slide here for the hands-on session that's coming up after the panel discussion, which we will move to right now. Okay.
Amazing. So now maybe we invite the other speakers. John, what do you think? Michael and Victor to join us here on.
Is Victor with us?
Victor is there in the last row, but I don't know if he wants to join here. Please, maybe whoever you want to sit.
I'll become Vic. We haven't had enough of you. We haven't got our fill yet.
Of course.
No, surely not. Okay. I think we can.
We always are.
Drop the slides now. Next slides. So questions, guys. Bring on the questions. Anything for particularly for those two, but also things that kind of span the themes that you've heard so far.